Hi, Micro Geniuses. It's Brian. I am so sorry I'm late with this video lecture for the review for Exam 3. Exam 3 is next week, and so I wanted to be able to go through these review questions with you directly. The exam, like normal, will be focused on these review points and those review points only. And so I just wanted to uh, make sure that you knew that. This is your best study guide. Um, you want to look up these particular terms and this information um, directly in your notes. And if you do that, then you're going to be in good shape. Okay, so here we go. What is a niche? A niche is if you look at an organism in a community, the niche is their employment. That's the role that they play in that particular community. Habitat is where the member of the community lives. A community, if you draw a boundary around a particular area and you look at everything that is living within that particular area, that comprises a community. So it's a community of mixed organisms, but it only includes living things. It does not include inanimate or non-living material. If you take that same box and you include in it living and non-living things, then we call that an ecosystem. Okay, so you can draw basically an arbitrary boundary around any type of space. Could be your desk, it could be your room, it could be your house, it could be your backyard. Anything that is living and non-living defines the ecosystem. And then the biome. A biome is a particular region, usually defined by latitude and altitude, and that is a characteristic region of what you would find in that particular latitude or altitude. Okay. Above biome is biosphere, which is all of the living material on Earth, and the atmosphere, the lithosphere, and the hydrosphere. Um, and then biome is one down, where again, you would find multiple ecosystems in a biome. And the biome is related to its altitude and its latitude, usually also its proximity to water. Okay. Uh, Number two, know what acid acetogenic bacteria produce, which is beneficial for plants. Acetogenic bacteria produce sulfuric acid. Okay, sulfuric acid is beneficial to plants because the plants can't uptake sulfur directly. They have to uptake sulfur as sulfate. Okay. You should know the topsoil, which is the top aerobic portion of the soil. The rhizosphere, which is the root zone of the soil where you would find plant roots. Mycorrhiza. Mycorrhiza are fungi that grow within the rhizosphere that attach to the plant roots and will scavenge available water as well as micronutrients for the plant roots. The vadose zone is the top zone of the soil, which is unsaturated with water. Okay, so if the pore spaces are unsaturated with water, we call it the vadose zone. And if the pore spaces are saturated with water, then we call it groundwater. Okay. Number four, stands alone. Know that methanogenic activity is ubiquitous in soil because of the abundance of CO2 released from limestone. So methanogenic bacteria breathe carbon dioxide. Okay, that is their terminal electron acceptor. So since there is such an abundance of carbon dioxide due to limestone in the soil, then you can count on finding methanogenic activity just about anywhere, as long as you're in the subsurface of the soil. Number five, you should know that toxic metal waste can never be fully be biodegraded because you're dealing with an atom that is toxic. So if you take something like mercury, you can never degrade mercury and make it into a non-mercury material. Okay, so it can't fully be biodegraded. It will be hazardous wherever it is, and you can't break it down further into something that's non-hazardous. The opposite of that would be something like gasoline, which is a molecule that can be broken down into carbon dioxide and water. But when you have a toxic metal, mercury, cadmium, zinc, lead, uranium, it cannot be fully degraded into something that's non-toxic. What we do in those instances with metallic toxic waste is we attach the waste to the soil and we minimize the amount that's dissolved in the groundwater. The groundwater will ultimately flow into different environments and will expose environments to these toxic metals. And this would include drinking water. So if we can bind the toxic metal to the soil, then it will be fixed in place, will not move with the water, and it will not be just a continuing contamination problem. Number 6, producers. When we're talking about chapter 24 here, producers are autotrophs. They can produce their carbon from or inorganic sources, and consumers are heterotrophs. They get their carbon only from organic carbon. Okay, so producers are always autotrophs. They can always make carbon compounds and organic compounds from CO2. Consumers, they have to consume organic carbon directly. Okay. Nitrification or nitrifying bacteria, this is number 7, is beneficial to plants because with nitrifying bacteria, which is the opposite of denitrifying bacteria, with nitrifying bacteria, then you have the ability to produce nitrate. Okay, nitrate is a plant fertilizer. It is beneficial to plants. And so nitrifying bacteria are beneficial to farmers and their crops. Okay. You should know what fecal coliforms are. Fecal coliforms are specific types of bacteria that are tested in municipal water. They denote the presence of feces in water or the contact that water has had with sewage. And so you do a special fecal coliform test on 100 milliliters of water, and if you see more than 4 fecal coliforms, then that water is unsafe for drinking. Should know the bacteria that cause impetigo. The first one is Staphylococcus aureus, and the second one is Streptococcus pyogenes. You should know that the protein that's most common in the outer layers of the epidermis is keratin. Keratin protein is impregnated in the stratum corneum. That's the dead layer of cells, and it's highly hydrophobic, and that makes your skin resistant to water penetration. Okay. You should know the viruses that cause smallpox. That would be variola. The virus that causes chickenpox is varicella. The virus that causes measles is morbillivirus. The virus that causes the common cold is rhinovirus. And the virus that causes rubella is rubevirus. You should also know what otitis media is. Otitis media is an ear infection. Usually it's an endogenous infection of Streptococcus pyogenes. And it is a sequela of a common cold. Okay, so sequela just means that it, quite often that it will follow the common cold. Okay, you should know the complications of strep throat. Okay, the complications of strep throat could include scarlet fever, rheumatic fever, glomerulonephritis. Glomerulonephritis is a disease of the kidneys. And then PANDAS, which is pediatric associated neurological disease associated with strep. Okay, so PANDAS. So again, the complications due to strep throat are scarlet fever, rheumatic fever, PANDAS, and glomerulonephritis. Okay, and the stage at which you have uncontrollable coughing in pertussis is called the catarrhal stage. Okay. And I have to say really quickly, oops, I knew I would get this wrong. The catarrhal stage of pertussis is marked by an uncontrollable cough. And the proximal stage, or I'm sorry, the catarrhal stage, oh my goodness, I'm getting this wrong. Catarrhal stage is just where you have cold-like symptoms. Okay, that happens first. And the proximal stage of whooping cough is when you have the uncontrollable cough. Okay, just so you know. Sorry about that. Okay. Okay, in terms of antigenic drift and antigenic shift, you should know the difference between these. Antigenic drift is when the virus mutates directly, when you have a different type of influenza virus and it mutates by just changing out bases within the genetic code. And antigenic shift allows the species to change on the flu. So you can have bird flu infect humans or swine flu infect humans. And that is just a mechanism where you start to share different hunks of nucleic acid, and you have then what is called antigenic shift, where you'll have a common host that might have two types of influenza, and then some of that DNA will be shared among the different viruses, and then that antigenic shift mutation will cause something that's not pathogenic to humans all of a sudden to become pathogenic to humans. That's called antigenic shift. Okay, so you only see antigenic shift when you see influenza go from one species to the next. Okay. To isolate fecal bacteria, you want to use what's called McConkey agar. McConkey agar is a type of agar where fecal bacteria, which are lactose fermenting bacteria, have a specific colony morphology where the colonies will appear pinkish and will have a pink ring around them. Okay. The medium to isolate Staphylococcus aureus is mannitol salts agar. We've used this before. Mannitol salts agar is used specifically for gram-positive bacteria, and Staphylococcus aureus, when it's isolated on mannitol salts agar, appears yellow. It turns the medium yellow, goes from pink to yellow. Okay. Should know the difference between a primary and a secondary immunodeficiency. Primary immunodeficiency you're born with, and a secondary immunodeficiency you acquire. So the prime example of a primary immunodeficiency is SCID. It's a genetic defect. SCID stands for severe combined immunodeficiency. And secondary immunodeficiency would be something like HIV, where you acquire that immunodeficiency after some period of time and you acquire it through some type of viral infection. You should know that secretory IgA are found in the lining of the respiratory tract. Okay, SIgA is a specialized type of antibody that will survive on mucous membranes and serous membranes. As far as the bacteria that make up the respiratory tract, you have Streptococcus pyogenes, Neisseria meningitidis, Streptococcus pneumoniae, and Staphylococcus aureus. Okay. I had to look on that one. See, I pulled up the chart for that. Okay. Effusion in terms of otitis media is buildup of pus and fluid behind the eardrum due to an inflammatory response. Okay. Middle ear infections are always endogenous infections. Okay, so you need to make sure that if you see a middle ear infection, that it is endogenous, it is from resident microbiota that's just trapped behind the eardrum. Pharyngitis is just a term that refers to inflammation of the pharynx, so sore throat. Rheumatic fever is a long-term sequela of pharyngitis, okay, specifically strep throat. So if you have strep throat, and that can lead to permanent damage of and long-term damage of the heart valve. Okay. The causative agent for whooping cough is Bordetella pertussis or B. pertussis. And the majority of the sore throats, the most common is Streptococcus pyogenes. Okay, the disease marked by uncontrollable coughing is whooping cough. And the best antibiotic for pertussis is azithromycin. The causative agent of the croup is respiratory syncytial virus. Okay, and then we take a bit of a departure here, and we're going to talk about environmental microbiology and bioremediation. So we're going to shift to that. So know what type of bacteria grow in near surface soils. So the closest to the surface, the more oxygen you have, so you would find aerobic bacteria. You should also know that sulfate-reducing bacteria neutralize sulfuric acid and form hydrogen sulfide gas. There's a test question on that. I don't know if I have. No, you know what? It doesn't appear that I have a point on that, but please know that sulfate-reducing bacteria will reduce sulfuric acid and turn it into hydrogen sulfide. Okay, biostimulation is where you add nutrients to the groundwater or the vadose zone in soil, and that stimulates normal bacteria, native bacteria that are already native to the soil. Bioaugmentation is when you add bacteria directly, so you're taking exogenous bacteria that are known to degrade the contaminants, and you're adding it directly to the soil or to the contaminated surface. Bioventing just means that you're adding oxygen via air to a contaminated site. Land farming is just when you actually tilling and turning soil so it gets greater aeration. Composting is when you build up soil and you amend soil with things like leaves and straw to increase aerobic activity. And monitored natural attenuation is when essentially you just monitor a site and you look at natural processes that degrade contaminants, and you look at the progress of contaminant degradation through the groundwater, through the vadose zone, to make sure that no contaminant is going to hit a place where it would be a major threat to human health or the environment. The best conditions, number 32, for degrading petroleum hydrocarbons is always aerobic conditions. Okay, and you also should know the degradation of perchloroethylene. Remember, perchloroethylene has 4 chlorine molecules on it. That is degraded to trichloroethylene, which has 3 chlorine molecules on it. That is degraded to dichloroethylene, that has 2 chlorine molecules on it. And then it is degraded to vinyl chloride with 1 chlorine molecule on it. Vinyl chloride is the most toxic compound in this degradation pathway, and so you must degrade vinyl chloride further by removing that last chlorine and getting ethylene. Okay, number 34, you should know that cyanobacteria are bacteria that during the daylight can photosynthesize and during night can fix nitrogen. So on a 12-hour cycle, they abolish all of their abilities to do photosynthesis at night, so they can do nitrogen reduction. And then during the day, they abolish all of their nitrogen reduction machinery and they recreate the machinery to do photosynthesis. Okay, in terms of aquifers, okay, in terms of water chemistry, oligotrophic means that there's only small amount of nutrition in the aquifer. Eutrophic means that there's a large amount of nutrition in the aquifer. Eutrophic aquifers usually are overgrown with algae, and the algae starves the lower layers of the water of oxygen, so things like fish will die. So an oligotrophic aquifer is usually in some type of stable equilibrium where the food web will survive, whereas eutrophic is more of a departure from equilibrium, where because of the algal blooms, then life below the algae, life below the surface of the aquifer will not exist. The common pathogens that contaminate water supplies would include Salmonella, which is a bacteria, Giardia, which is a protozoa, Vibrio, which is a bacteria, hepatitis A, which is a virus, Norwalk, which is a virus, Shigella, which is bacteria, and Cryptosporidium, which is a protozoan. Okay, so those are 7 of common pathogens that contaminate water supply. Again, we have Salmonella, Vibrio, and Shigella, which are bacteria, Giardia and Cryptosporidium, which are protozoan, and hepatitis A and Norwalk, which are viruses. Okay, you should know that Bacillus thuringiensis produces a natural insecticide. Bt crystals are just made from Bt bacteria, and that can be sprinkled directly on plants to make a natural pesticide. Okay, now let's talk about rashes. Yay, rashes. Okay, so you should know the difference between a macule and a papule. Okay. A macule is a discoloration, like a freckle or a red mark. Papule is a raised bump. Okay, so if you have a maculopapular rash, that means that you have not only do you have discoloration, freckles, or red spots, but you also have raised bumps. A lesion is just a site of disease. Okay, a vesicle is a larger portion that is filled with fluid, like a blister. A pustule is a larger portion that is filled with pus. A plaque is a large, like a hive. It's a large, has a flat top to it, and it's usually larger than 1 centimeter. A purpura is more of a kind of a purple mark that is looks more like a spider web. And a cyst is a walled off infection that is usually hard when palpated. So if you try to squeeze it or anything, it remains its hard texture. You're not able to pop it. Okay. The causative agent for scarlet fever comes from strep throat. So you'd expect Streptococcus pyogenes. The organism that causes ringworm is a fungus. Okay, so ringworm is not a worm, it's a fungus. You don't need to know the specific type of fungus, but please do know that it's fungal in nature. The first vaccine that was developed by Edward Jenner, and it was developed for smallpox. Gas gangrene is caused primarily by Clostridium perfringens. Tetanus is caused by Clostridium tetani. Okay, in terms of impetigo with a lysogenic phage, that, remember, impetigo can be caused by Staphylococcus aureus or it can be caused by Streptococcus pyogenes. But the bacteria that causes impetigo that has a lysogenic phage in it is Staphylococcus aureus, not Streptococcus pyogenes. So Staphylococcus aureus, when it's infected with a lysogenic phage, can cause impetigo. Okay. The different proteins listed here, protein M, streptokinase, coagulase, elastase, and the collagenase, are different proteins that are active in impetigo. Okay. The protein M, well, let's go back and look at it. Okay. Protein M attracts plasminogen. Okay, it has a high affinity for plasminogen, and plasminogen is activated by streptokinase. Plasmin then degrades tissues by digesting fibrin and other tissue proteins, and collagenase directly is caused by Staphylococcus aureus. So you have coagulase, which coagulates plasma and blood, hyaluronidase, which digests intracellular glue, streptokinase that dissolves blood clots, and these are all in Staphylococcus aureus. The causative agent of warts is HPV, human papillomavirus, and you should know the layers of the epidermis. The top layer of the epidermis is called the stratum corneum. Below that is the stratum basale, And then the basement membrane is below that. And then below that is the subcutaneous layer, which is the dermis. Okay, and then finally the muscle layer. And that concludes this video lecture. If you have any specific questions on the study guide, make sure to email me directly and I will provide as much guidance as possible.